Module 4.3 Implementation and Applications of Coarse Graining Techniques. Note this is especially applicable for soft matter such as polymer, proteins, and so on. We're going to briefly discuss what we learned in the previous modules about coarse graining. We'll start with a simple toy example with 4 atoms and see how we can apply coarse graining techniques. Then we'll go into more detailed problem with an explicit protein example, and we will learn how to prepare the structure, how to set up the coarse graining beads, how to set up the force field solvation model, and then optimize the structure. And then we will learn about the post-processing techniques that we can get the details about that minimization process. So we learned about this in the previous module as well, but just to refresh, there are different types of coarse graining. So you can do structure-based coarse graining or functional-based coarse graining. Typically, we're dealing with structure-based coarse graining. We want to select the coarse graining model depending on the problem you want to solve. So definitely discuss how to choose appropriate level of coarsening based on the system and the goals of simulations. And we also discussed previously about different types of software and tools that are available out there. So some of the most common one that are listed here, like Martini, Gromax. So we're going to use those softwares for the tutorial session. And yes, based on the problem you want to select or solve the problem, you want to select appropriate software. So let's start with super basic toy example. So in general, this is my advice that anytime you want to model a system with different modeling method, start with some toy problem, something you can comprehend and analyze in more details. And once you have a basic grasp of what is going on, then I think it's easier to extend that to more complicated problems. So always start with toy problem if possible. So in this toy example, we are showing you a 4-atom system. So atom 1, atom 2, atom 3, and atom 4 given by their x, y, z coordinates. And then we define Martini beads where the bead 1 is a combination of atom 1 and atom 2, and bead 2 is a combination of atom 3 and atom 4. So one of the way to map them, remember we talked about the mapping function chi. So here we map them based on their average or mean position. So here we are creating beats, so beats in a dictionary here, and then we are calculating the average position of atom 1, atom 2 as bead 1, and atom 3 and atom 4 as bead 2. And then we can see on the bottom here, the bead 1 is an array which 1.5, 2.5, 3.5 x, y, z coordinates, which is just the average of 1, 2, 2, 3, and 3, 4, and so on. So now, anytime we perform modeling technique, we first visualize this. And this is very useful for getting some intuitive idea. So let's visualize this very simple 4-atom system and the coarse grain 2-bead system. So here we are going to use the Pi3D-Mol. So 3D-Mol is a very popular molecular visualization package. So Pi3D-Mol is just a Python wrapper for that. And we will be using both the beads as well as the atomic structure. So in the atoms shown here as the red spheres, and the beads are shown as the big blue spheres. And you can see we have changed the visualization from radius 0.3 to 0.5 to maybe make the beads look bigger. Yeah. Now let's start with very simple Fene model. We talked about that in the earlier slide. So, but here, instead of dealing with 3D problem, we start with a very simple 1D problem. So we need R, the position vectors. So here, because it's a one-dimensional case, we'll start with 0 to 1.5 non-dimensional extension of the system we want to study. And then we make a line space that is a grid of 100. And then we give it to the Fene model, the def, the function we wrote in Python, which 2 parameters, k and r0, and this might change based on the problem you want to solve. And in this way, I can show you the plot on the right, how the based on the distance, we can get different Fene potential. Now we can move beyond the 1D model and have the beat position as 1.5, 2.5, 3.5, and so on. And there you can see that we are setting the Fene parameters, and then there's an important function here, NP dot linalg dot norm beats position 1 minus beats position 0. So it's basically calculating the distance between them. So instead of dealing with r, which is the coordinate of the beats, we are dealing with rij vector, which is the distance vector. So we can plot how does the beat position change over time on the right-hand plot. Now let's deal with a more realistic example of this. So one of the most popular application structure for coarse graining, as we discussed earlier, are proteins. So we start with one of the protein structures. So first of all, similar to other crystal structure databases like Jarvis, DFT, Materials Project, OQMD, crystallographic open databases, there's different protein databases called, and one of the most famous one is PDB, or Protein Data Bank. So it has around several 100,000, 220,000 plus structures of PDB. So a lot of people over the years have kind of submitted their structures here. So thanks to all of them, all the wonderful scientists out there who are continuously uploading their protein structure files so that people can download, analyze. And in fact, some of the COVID vaccine proteins are also uploaded recently here, and there are a lot of research work based on this protein data bank. This is a very popular website. So we start with 1AKI example protein, which is a hen egg white lysozyme protein. We don't need to go in the details of biology of the system and stuff, but what we need is the crystal structure or coordinates. So we download this 1AKI crystal structure using the wget command, and then sometimes we do not want to simulate hydrogen atom, so or some unnecessary atoms. So you can see in the grep command, we are cleaning the 1AKI.pdb to 1AKI clean.pdb. And then we can, similar to what we did in the previous case of visualizing 2 beads, here we'll be visualizing the 1AKI bead or 1AKI clean.pdb using the Py3DMOL software. Now, similar to what we performed the coarse staining in the previous 4 atom case, where we took the average of their position to get the center of mass, here we'll be using Martinez software, which has nicely automated the coarse graining process. And there are several hyperparameters, but let's start with the Martinez 2 minus F is the file input, which we just prepared in the previous slide, 1AKI clean.pdb. And we want to output 1AKI CG, coarse graining PDB, also the topology file -o topol.top, and what kind of force field we want to use. There are different force field. So we're going to use Martini 3001 and so on. So Martinez automatically converts the fully atomistic model to Crane's coarse grain model. So you also want to set up the force field, so we can get for the force field from one of the website here. We can untar it. So untarring is basically the way to unzip in a way, and then we can visualize one of the content of this file on the right hand side. So these are basically done using VI. VI is an editor in Linux. Some of you might be familiar with. So VI file name will open the VI. And then you can see what are the content of this particular force field. So there are force field for solvent, ions, and so on. So you can visualize them as well. We won't need that kind of detailed, sophisticated information, but it's just good to know what is out there in the file before we start running it blindly. Now, the important step, we're going to minimize the structure or optimize the structure. So remember, those proteins that are uploaded in the protein data bank are maybe from experiments or some sort of refinement, but they may not be suitable for the particular physics that we want to capture. So we want to optimize the structure using our force field, using coarse graining or using fully atomistic models. So here is the case for coarse graining technique. So we are doing GMX-MD run, so which is GMX is the GROMACS command here. And then we'll be using the def NMEM command, which is basically where the output will be. So EM is energy minimization. And then we produces several outputs such as em.log, For the detailed information about the run, em.edr, the file containing energy terms, em.trr, trajectory file containing the position, velocity, forces of the atoms over the minimization run, and .grow is the final configuration, which is the GROMACS file. So one thing to highlight here, you can see the blue box here, the energy is going down. So minus 1.73 to minus 2.22 to the power 10 to the power 5. So basically energy should go down. So the idea is the lower the energy, the more stable the structure. And same with the forces. The lower the forces, the better. So after the run, we can post-process the files. So in GROMACS, there are post-processing tools. So here we are giving the 9th entry. So there you can see the 28 entry on the bottom. Like you want to visualize bond, virial stress, pressure, Coulombic interaction, Lennard-Jones interaction term, and so on. So we can visualize, for example, potential here. So this could be useful to realize whether my energy is being minimized or not. So which is definitely the case here. On the right-hand side, you can see energy is going down. That means we are trying to get more and more stable structure.